Have you ever considered the life of a fish and the challenges it faces? You're swimming in the sea, minding your own business, when suddenly a bigger fish comes out of nowhere trying to eat you. You manage to escape, only to find yourself tempted by the most beautiful piece of fishing bait on a fishing hook. After restraining yourself, you find yourself caught in a fishing net. It's a stressful life. As humans, when we get stressed, our body produces a stress hormone, cortisol. Cortisol is what allows us to respond to our stressors and still function. But what about these fish? Those are Pacific hagfishes. The Pacific hagfish is a marine fish considered key in the evolution of vertebrates. However, its stress response is very poorly understood. We do know that hagfishes produce a transparent mucus-like slime when they get stressed. But my research looks more deeply at how hagfishes respond to stress. This includes investigating what their stress hormone is, where it is made, and how it is produced. Let's consider human stress for a moment. When an animal like you or me experiences a stressor, the stress axis activates a whole cascade of events in our bodies. First, there's a small region in our brain called the hypothalamus that releases a hormone to the pituitary gland when it perceives a stressor, either inside our bodies or externally. At the pituitary gland, that's also in our brain, another hormone is released, and that hormone travels all the way down to our kidneys. Atop our kidneys are two more glands where yet another signal is received and another hormone is released. That hormone is cortisol. Cortisol then travels via our bloodstream to different tissues to cause other events. Now, if we want to understand a bit more about how the hagfish responds to stress, well, what I did is that I injected some hagfishes with a stress hormone to accelerate their physiological response, while others I did not inject. Then I subjected them to a range of different stressors, because different kinds of stressors can cause different effects and different hormones to be produced in their blood. Then I collected blood, gill, and kidney samples. So with the blood samples, I'm analyzing them for any stress hormones present, and the tissue samples, I'm extracting RNA and looking if there are any structural changes. From this, I hope to identify probable stress hormones and those enzymes involved in creating those hormones. It is possible that hagfishes may use pregnenolone or progesterone as their stress hormone. However, it's a little too early to say for certain. My research will establish the stress hormone of the Pacific hagfish and allow a much broader understanding of the stress axis of more primitive vertebrates. And we can know how fish like these manage their stressful situations just like us.